This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 541, recorded on March 29th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello there, Vincent.
Been out lately?
Just got back from outside.
Is it nice out?
It's a little bit crisp. It's a little, it's spring.
It's 14. It's 14C.
It's spring. And it's a little drizzly.
Really?
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 11C here and overcast and rainy. And for some reason, the cat is yowling at the office door. But,
really?
He wants to have a say in viruses.
Wants to comment on the virus.
You want to talk about feline viruses?
I don't think they're on the agenda for today, but we will eventually.
No.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's sunny and 53 degrees Fahrenheit. And I can't get it to convert quickly, but it's pretty nice.
That's about 11, 12, something like that.
If you like what you hear here on TWiV, consider supporting us financially. Go to microbe.tv/contribute for a few ways you can do that, including give it $1 a month to Patreon or PayPal. We'd really appreciate it. And more than that, we could use it.
Indeed.
We have some expenses. And we would like to, as they say, grow.
Indeed.
The podcast. Indeed. Oh, today we have two cool stories again, as usual. We're going to talk about mitochondrial viruses, and we're going to talk about quasi-enveloped viruses. Well, what could be better? Sit back, relax.
Nothing.
Pour yourself your favorite beverage.
Exactly.
Not if you're driving.
No.
I guess you can't sit back and relax all the time.
No.
I guess people who sit in a nice, easy chair in front of a fire are not going to listen to podcasts, right?
And that probably not.
I don't know. They might.
Now, many years ago, 2010, I actually went back and looked this up. Someone wrote us a letter on TWiV and said, is there a virus that infects mitochondria? It's an organelle with its own genetic material and polymerase, so it could replicate a virus, or are the membranes a barrier? And I remember we said, no, there aren't any. Because I looked it up and I didn't find anything. It was 2010. It's only 9 years ago.
That's all.
But
they do exist. And how did I realize this? Well, we were doing a paper of Eugene Koonen's not too long ago where, remember that one where they looked at the phylogeny of all the RNA viruses over time? And he said, there are mitoviruses. Oh my gosh. They're actually mitochondrial infecting viruses. So I've been meaning for some time to talk about them because that's very cool. Mitoviruses, right? Mitochondrial infecting viruses. And they belong to a family called the Narnaviridae, which Alan and I said before remind us of the Chronicles of Narnia.
Mm-hmm.
Every time I hear that. So there are mitoviruses and Narnaviruses in that family. And they're both very similar, except the mitoviruses replicate in mitochondria and the Narna replicate in the cytoplasm. But otherwise, they're similar. They're plus-stranded RNA viruses. They are pretty small genomes between 2 and 4 kb, and they encode only one protein, an RNA polymerase. No capsid protein. These are capsid-less viruses, which should tell you right away they don't go outside of the cell. They stay inside. They listen to podcasts.
Weird.
Right. They sit by the fireplace.
It's weird, isn't it?
The powerhouse of the cell. So
Exactly.
they are sitting by the fire.
Exactly.
Oh, and they get plenty of energy.
Yeah.
Is there energy inside the mitochondria?
Well, it's producing it.
And it
The
comes
electron
out.
transport chain and all that. So it's generating ATP,
I
right?
thought it was in the membrane, and then the ATP goes out. It doesn't actually. I guess so. Okay.
Right. I mean, it can replicate viruses.
Yeah. So they have their own tiny genome, but more importantly, they can do protein synthesis, right? In there, they have their own set of protein synthesizing machinery.
And these are found up until the paper we're about to talk about. These had only been found in fungi, right?
That's right. They were found in the 80s in 2 different fungi, the Dutch elm disease fungus.
Really?
Ophiosoma ulmi, and the chestnut blight fungus.
2 very important fungi.
Wow.
Griphonectria parasitica. And
so do they actually kill the...
no, they don't.
That's too
they
bad.
mostly do nothing. Yeah. They're called cryptic viruses.
Cryptic.
So, Dickson, can it be a virus without a capsid? Sure.
Apparently.
they don't make the fungus sick, and sometimes they make it less virulent, because these are, of course, pathogens of trees, right?
Yes.
And so apparently if you have the virus in the fungus, it makes it less pathogenic. And there are other fungi that are known to do that as well. And there are like lots of fungal mitovirus genome sequences in the database.
Oh, interesting.
Yeah, they're over 100 or nearly 100.
Wow.
So Vincent, I think it was Vincent, included a background paper for us, and I didn't get very far into it, but I want to report one sentence. Mm-hmm. The initial description of mitoviruses was accomplished through research on 2 related fungal pathosystems that are often confused by those with only passing familiarity in plant pathology.
Us.
Yeah. And so one is the Dutch elm disease fungus, and the other is the chestnut blight fungus. But I just thought it was so funny. Often confused by those with only passing familiarity in plant pathology.
Every morning when I wake up, I have to read the inscriptions on my wrist that I wrote with a pen the night before to make sure I'm not going to mix these up.
And, they were first discovered because they found double-stranded RNA in these fungi. And even though these are single-stranded RNA viruses, that's the replicate of intermediate.
Ah, right.
So the only viral protein, it's interesting, is this RNA polymerase, which is made in the mitochondria. It copies the plus into a double-stranded RNA, and then that's copied to more plus RNAs. And no capsid. And even the NARNA viruses in the cytoplasm, they don't encode capsids either. They just stay there. So as the cells divide, they divide with them. And sometimes fungi fuse, right? Anastomosis.
Right.
And so they can go from
they form a syncytium.
Cell to cell that way. Yeah. So I guess you should never rule out anywhere. And I understand chloroplasts have viruses too. Maybe we'll do that another time.
Well, if mitochondria can get them, why not chloroplasts?
Yeah, they should.
Now, I picked the paper just for us to peruse for a couple of reasons. It's a recent paper on mitoviruses.
So wait a minute. Mitoviruses are green with envy at the ones that infect chloroplasts?
Yes. Yeah, that's right. Let me just say one thing first, though, before we do this paper. In that paper by Kunin, they said mitoviruses are very close to a bacteriophage, leviviruses. These are filamentous single-stranded RNA bacteriophages. Think about this. Mitochondria are former bacteria that invaded a cell a long time ago and stayed there, right? So maybe when they invaded, the leviviruses came with them, and leviviruses encode capsids. And then they said, we don't want to leave. Let's stay here. And they
so the levy broke.
The levy broke, and they lost their capsid, and then they remained as what we now know as mitoviruses.
They're homeless viruses.
They lost the capsid because now when they traveled between cells to them, which were now mitochondria, there was no need for the capsid because they were all in the same.
That's right. They divide, they replicate, and as the mitochondria go from cell to cell. Now, what was that? These are old. On the phylogenetic tree of RNA viruses, they're close to the root. So this is consistent with the mitos originating from leviviruses. The levy broke. This paper is called Evidence for Contemporary Plant Mitoviruses. And the authors are Max Niebert, how do you say, Niebert or Niebert?
Niebert.
Niebert.
That's how I've heard it.
Minvong, Karen Fugate, and Humberto Debat. So Max is at Harvard Medical School, and I've known Max for many years as a Rio virologist. He trained with Bernie Fields originally. And he went off to do a postdoc, then came back, and he's gotten interested in exotic viruses like mitoviruses. He works on the viruses that infect Giardia.
Oh, yeah.
The Giardians of the galaxy.
Right. I liked that one.
Those are double-stranded RNA viruses, aren't they?
I think so, yeah. Yes, they are. And so here he's got this paper, and also there is contribution from the Agricultural Research Service in Fargo. Oh, boy. Someone from Fargo.
Yeah.
And Argentina. Instituto Nacional de Tecnología Agropecuaria. My former postdoc came from there.
In Cordoba, Argentina.
INTA. So yes.
I was just going to say, because the title has this evidence for contemporary plant mitoviruses, in the first line of the discussion, it almost seems like the reviewers said, what do you mean by contemporary? Because they say, parenthetically, modern day extant. So that's what they mean by contemporary. These are existing and they're current.
Because it could have other meanings, right? Like my contemporary desk here, Dickson,
It was
right?
a modern virus. That's right.
Right.
Modern virus. So what they did here is...
I think most viruses are built more in the brutalist style.
They took a look through the transcriptome shotgun assembly database for lamb plants. It's the other TSA. So this is public sequence, and they searched it with the mitovirus. And they found, in the end, after a lot of machinations that we don't need to go into, they found 20 nearly complete genome sequences of apparent mitoviruses from plants, 10 different plant species, and same sort of length, and a single open reading frame that encodes a protein that looks like an RNA polymerase.
Wow.
Well, actually, did you say 20? Because I thought they narrowed it, or you said 10. Okay. Okay.
20,
and then they're going to get to 20 eventually by using this other database, the sequence read archive database of DNA sequences.
Right. Okay. And when they're doing this, they're looking at so they're looking at sequences of things that are actually transcribed that are RNA.
That's right.
Which is important because there are these endogenized RNA virus elements, non-retroviral endogenized RNA virus elements, or NERVs, which show up in the in the genomes of plants.
That didn't show up in any of our potential titles. I figured it would.
No, we didn't put any nerves in there.
Viruses with nerves.
And so I may have misspoken. The sequence read archive database, that might be transcriptional data as well. Yes, I think it is.
So.
So in the end, they have 20 refined sequences from 10 different species of plants, right? Mm-hmm. Okay. And Dickson, you're not going to ask what kind of plants?
Angiosperms or gymnosperms?
Beats me.
Beets. Corny.
Yes, beets, potato, and
Tunia,
dahlia,
fern.
Fern.
So the fern is the outlier. Yep.
And cannabis sativa.
Yes, right.
Hemp.
Yes.
So first of all, you could say, well, how do you know these aren't from fungi that are either symbiotic to the plant or contaminating, right?
So how did they know?
Well, they did a phylogenetic clustering, and they seem to be monophyletic, that is, deriving from a common ancestor. And if they were from fungi, they would be all over the place. So probably they're not from fungi. But an even better piece of evidence is something that I did not know. Did you know that?
It's really cool.
You know that in fungi, UGA, which is normally a termination codon everywhere else in the world, in the mitochondria can be translated to tryptophan.
Really?
I didn't know that.
Huh.
So mitoviruses of fungi have a number of UGA codons throughout the coding region that don't stop translation. Tryptophan is inserted. And these plant mitoviruses...
But only when they're in the mitochondria.
Only in the mitochondria, of course. Right, because in the cytosol, like the narnoviruses don't have these scattered UGA
Which
codons.
would be a way to keep them from replicating in the cytoplasm.
Yeah. What? I don't know why these
it's
would
just
evolve.
weird.
It's
Fungi
weird.
are just weird.
So anyway, the plant mitoviruses do not have internal UGA codons, which is another piece of evidence that they're not fungal. They're
higher fungi or lower fungi? He asked.
Well, yeast and
that's a lower fungus.
There's some filament.
And the Dutch elm disease and chestnut blight.
Any basidiomycetes?
We don't know.
I don't know offhand.
I don't think so.
Okay.
It's only in some fungi that this is a rare mitochondrial codon.
Yeah, it's not in all
Right. So
of
it's a couple of Saccharomyces species, Cryphonectria and Ophiostoma. And those are the only ones I think that are mentioned in the paper. So yeah, it's I don't think any of those are basidiomycetes, are they?
No.
Mitoviruses were pretty much the same throughout each plant, which you would expect for mitochondrial viruses. They would be pretty much the same throughout all the tissues of the plant. So that's really all I wanted to say about this, but we don't actually know if these are replicating in plants.
They did do one experiment.
They did.
This is almost entirely an in silico paper, but there's a validation of one of these.
Oh, that's right. Yeah.
Where they let's see, they isolated it.
They isolated RNA from leaves.
Isolated RNA from leaves and seeds of beet plants.
Mm-hmm.
It's not exactly a plaque assay, but they do show that you can find this RNA in actual living plants and amplify it out by RT-PCR, and it's the right size and contiguous and could be translated, and so it suggests that this is actually
okay.
And you imply that they do no harm, so the beat goes on.
Do no harm.
Yes.
When they look at the tissues, they find some evidence for higher frequency of the virus maybe in flower-related samples. So that includes seeds as well. That's interesting. Tenfold higher, more in the petunia, for example, in the flowers.
I found another paper, which was published actually this year, where they develop a method for introducing mitovirus RNA into cells using protoplast fusion. So you can start doing experiments, and you can show that it gets in the mitochondria and replicates. So you could do, I guess, is that in plants? Let me see. Yeah, I think it is. Hold on. Let me just make sure here. So it's just a general virology paper. Cryphonectria parasitica is a fungus.
Right.
So they figured out a way to put the RNA back into the fungus and get it to replicate. So that's good. So you can do some experiments, but they'll have to do that with plants. Show, if we introduce the RNA, it can replicate.
Right. I guess you'd have to have some plants that don't
have it. Yeah. Or put a different one in. I wonder how specific they are to the plant, right?
That would be an experiment.
Yeah, or the fungus, if only certain ones going to replicate in a fungus.
Right.
Species, and what controls that would all be interesting, right? Good luck getting that funded.
And
from the
good
ag
luck
department.
Getting that to work. I guess the Saccharomyces ones would be the ones to start with, because you can culture those easily in the lab.
Yeah.
Oh, good. The Mueller report's coming out.
And then you'd have the awesome power of yeast as well.
Yeah.
So mitochondria can't be cultured by themselves, but can they be cultured in an enriched cell-free extract?
I am not aware of that.
I'm not either, but I'm just asking because things have happened since I last followed that literature.
Oh, no, nothing's happened. No, they're highly reduced genomes. They can't do anything on their own.
No,
but you could imagine that what Dickson's suggesting is that you might have a very in vitro system of just the mitochondria.
Correct.
I mean,
but
people must have tried that.
I'm sure they're trying it as we speak, but because they do replicate, obviously, and so therefore they look like they're independent of something. They don't replicate just because the cell replicates, right?
A lot of that electron transport machinery is from the nuclear genome, right?
I
Yeah.
know that.
So that would be, they might apoptose rapidly. But you can put them in other cells.
You can.
Yeah. You can take them out of one cell and put them into another.
Exactly.
Just think if you had huge farms of mitochondria, you could make energy.
The thoughts have occurred to me, but only in...
That's the idea behind the Matrix. They took farms of humans and...
Farming humans. Not a very efficient way to do things.
No.
But
they
made for a good plot, at least in the first movie.
Right.
The rest were horrible, weren't they?
That was all downhill. Yeah,
exactly.
So those are mitoviruses. We now corrected a 9-year-old error on TWiV.
There must be plenty more out there.
Oh, yeah. We have lots of errors.
And we're adding more as we speak.
I take full responsibility.
Mitochondria are really interesting.
I think you're right.
I read a whole entire book on mitochondria.
Oh, yeah. And who was the champion for mitochondria? Leninger.
He was?
Yeah. Absolutely.
Let me tell you the name of this book about mitochondria. It's really good. Mm-hmm. It's by Nick Lane. Here it is: Power, Sex, Suicide, Mitochondria in the Meaning of Life. I've read it. It's really good. And then he has another one on oxygen, the molecule that made the world. Right. That's really good.
Right.
He writes very well. He's a biochemist in the UK. Okay, our paper is also really interesting, and it takes up something we talked about a while ago. This is from eLife, which is the journal where you don't have to do additional experiments. Our plant paper of a few weeks ago was in eLife. Cellular entry and uncoating of naked and quasi-enveloped human hepatoviruses by Rivera, Serrano, Gonzalez, Lopez, Das, and Stan Lemon, University of North Carolina at Chapel Hill. And hepatitis A virus is an unusual picornavirus, which of course is the family that contains the polioviruses that Alan and I have worked on.
Indeed.
And these are classic naked icosahedral viruses, right? They are composed of a protein shell, which is sitting right in front of you on the desk there, Dickson. No envelope. And then there are viruses with envelopes like influenza viruses and many others. And, historically, these two have been separate. But not too long ago, Stan Lemon showed that there is a quasi-enveloped form of hepatitis A virus. They're released from cells in small extracellular vesicles, no viral proteins on the surface, and a couple of 2 or 3 capsid per vesicle. They seem to be produced by the exosome machinery. And since then, other viruses have been shown to do this as well, like poliovirus, Coxsackie virus, which are all picornas, also hepatitis E virus, rotaviruses, and noroviruses.
So then a couple of immediate questions are, first of all, is this just an accident? Is this some kind of dead end that happens occasionally and has no impact on the virus, or does it serve some role? And secondly, how the heck are they going to get into their next cell if they're tucked up into an exosome?
And so Lemon has suggested that these quasi-envelopes would shield the particles from neutralizing antibodies.
Yep.
So that's
a cloaking device.
Cloaking device, exactly. And
so that's the benefit, right?
Yeah.
And maybe if you want to put more genomes in a cell, that would do it. And we did a paper on that. So I think we did Stan's original paper on this. And then we also did a paper showing that if you have more particles, you get better infection or something like that. So there are a couple of possible benefits.
So then the question is, how are they going to get into the cell?
Right. Now, when we were on, well, first let me say that where do you find these viruses? Okay. So in feces of people, so hepatitis A is an enteric infection that's passed by fecal oral contamination from one person to another. And it gets into your intestine and somehow makes its way to the liver, where it replicates and makes you jaundiced, causes hepatitis. The virus in feces is naked, right? The virus in the blood of these people is quasi-enveloped. And if you grow virus in cell culture, it is quasi-enveloped. It's interesting, isn't it?
Yeah.
And these envelope viruses are infectious. They're a bit different. The virus inside is a bit different from naked virus in that the capsid protein VP1 is not processed. Normally, there's a proteolytic site that's cleaved in the mature particle, but in the quasi-envelope, it's not. But it doesn't seem to matter. It's still very infectious. And these are made in exosomes, the little membranes that are shed from cells, and they depend on the escort machinery to do that. Okay, so when we were in Iowa a couple of weeks ago, we talked a little bit about TIM-1, T-cell immunoglobulin and mucin-containing domain 1 protein, which is a receptor for phosphatidylserine, which can be on the surface of membranes. And my former postdoc, Gerardo Kaplan from INTA in Argentina, when he left and set up his own lab, he identified TIM-1, and he called it the receptor for hepatitis A virus. Now, this is way before we even knew about quasi-enveloped hepatitis A virus. It turns out that TIM-1 is known to bind phosphatidylserine. That's what it's binding on these quasi-enveloped viruses, because there's no viral protein in the quasi-envelope, but there is phosphatidylserine, and that's what's binding TIM-1. And that's how it attaches to the cell surface. And Wendy Morey told us about that. However, TIM-1 is not essential for attachment of either Hep A, either with or without an envelope. It's not needed in mice either, so there's must be some other receptor that is needed for both viruses, for naked or quasi-enveloped. I just like that, quasi-enveloped. And it's quasi because it's not a traditional envelope like influenza virus or HIV or any other envelope virus.
So how related are the hepatitis viruses, A, B, C, D, and E?
Well, A is a coronavirus.
Not very.
B is a DNA virus.
No, I knew this.
C is a different plus. Yeah, right. They're all very distinct.
So when you say hepatitis virus, it's
right.
It's confusing a little bit because
this is the problem of naming viruses after common pathologies.
Exactly.
Because they were discovered that way, and this virus causes hepatitis. This virus causes hepatitis, but it's not the other one. And so they just started lettering them, thinking, well, these viruses have this in common, and that was all we knew they had in common. And then we end up discovering that this is a common endpoint of radically different viral life cycle. And hepatitis A it's not just the fundamental biology of them; it's the etiology. Hepatitis A is spread orally. Hepatitis B is the bloodborne, and it's just
et cetera, et cetera.
We're stuck with these.
I did an interview with Stan Lemon, TWiV 389. I called it Alphabet Hepatitis with Stan Lemon because he knows all about. He's a doc. Kind of a doc, is he? ID probably, but his speciality is hepatitis, so he can talk about all of them.
Isn't the liver the largest single organ in the body?
Skin.
Well, besides skin. Okay, I said in the body, not on the body.
Oh, come on, Dickson. You're being pedantic.
Yes, I am.
Right. Probably second to the skin, right?
Yeah, it's huge.
It's not bigger than brain, right?
No, but it's a big target. It's not bigger than muscle, but that's spread throughout the body.
Blood goes right through it, right? So if you inject something into intravenous, it doesn't take long before it gets to the liver.
Yeah, because it detoxifies stuff.
But so these viruses can infect liver for various reasons.
Well, if they're enteroviruses to begin with, that's the first place you go.
Well, they're not, and they're enterically transmitted. But enterovirus is a genus. Be careful.
I meant enteric.
Yeah,
in the sense of root.
Yeah, we did.
Do all? No, that's true. I back off.
So
now the edge of my knowledge.
So this paper is all about how. So we don't know what the receptors are for these viruses, okay? But this paper says, well, after the virus is bound and taken up, how does it pass through the cell? Are there any differences, right? And there are differences, as we will see, that are quite interesting. It's a very beautiful paper with lovely experiments.
Right.
And,
I would put this in the category of they probably would not have had to ask for more experiments.
No, absolutely.
Right.
Yeah, absolutely. Because it's very complete.
Thorough.
It's very complete. So when viruses are taken into cells, if they don't fuse or enter at the plasma membrane, then typically they're taken in by some kind of endocytosis, and there are many flavors of endocytosis, right? So you can say which endocytic route does the virus take in a number of ways. You can use inhibitors that are specific for each route. You can use antibodies against proteins that are specific for each route. For example, there's clathrin and dynamin dependent endocytosis. There's caveoli-dependent endocytosis, and they have different markers, and they go to different
Which sounds like
places.
an Italian dessert.
Caveoli.
Does all the cavioli, please? But the
Oh, caveoli. Now there's the pasta, which is cavatelli.
cavatelli. That's right.
The caveoli. Then
Sounds
there's
like a white wine.
Then there's
Could be
the
a white wine.
Right?
cannoli. There's
I've
cannoli.
endocytosed a few of those.
Cannoli dependent.
I've exocytosed a few of those.
Yesterday we did a paper on TWiV about the ear, and there are all these Italian structures in there. The scala timpani. I was going crazy with the Scala. And you know, La Scala is the opera house in Milan. Does anyone know what Scala means?
Scale?
Opera house?
Opera house in Italian. Scala.
I thought it meant a room, but
Well, I don't know, but I've toured La Scala.
yeah.
Ah.
No, but anyway, Elio said it wasn't room because Scala timpani. It kind of, it's the inner ear has these different chambers.
Plays the Scala timpani very well.
So anyway,
Ladder.
there are these different endocytic pathways.
Right. So they said which
Scala
one?
means ladder.
Ladder. Thank you.
Ladder.
Oh, okay. Right. I wonder why the ear is Scala.
Yeah.
Anyway, so they used these drugs and antibodies to show that both non-enveloped and enveloped hepatitis A basically are using clathrin and dynamin-dependent endocytosis. Because there's, the caveolin may play a little bit of a role, but mostly it's clathrin-dependent, and that's pretty clear, okay? Now, the next they say, let's look at surface molecules that might be involved. They look at integrins, which are surface molecules, or adhesion molecules involved in cell-to-cell adhesion. And in fact, if they knock down the production of one integrin called integrin beta 1, they get a pretty good reduction in the uptake of both quasi-enveloped and naked viruses. And they also do a knockout with CRISPR, and they can throw a peptide of 3 amino acids, which is an integrin binding motif, which reduces uptake. So it looks like there's somehow surface integrin beta 1 is involved in the uptake. It's not a receptor, but it's somehow involved in the uptake. And they do see some differences in the interaction of the two viruses. Again, the naked and the quasi-enveloped in beta 1, but they don't know what that is. Okay, so integrin beta 1, but what it's exactly doing is not clear. It's just involved in the uptake. All right, next is an interesting series of experiments. There are GTPases involved. These are proteins that they hydrolyze GTP. They're involved in sorting of cargo through different kinds of endosomes, early versus late. So as something is taken in from the plasma membrane, first it's put into an early endosome, it becomes late endosome as it proceeds towards the nucleus, the pH drops, and eventually they fuse with lysosomes. That way you can release things that have been brought into the cell. So depending on what kind of GTPase, it can be an early or late endosome, and they have ways to look at that. They can look at antibodies, for example, and say, where's the virus? Is it an early or late endosome? And so they can see that both the naked and the quasi-enveloped clear and early endosomes about 1 to 2 hours. They're not associated with what are called recycling endosomes. And they end up getting into late endosomes as well. And they can deplete these GTPases and show that it affects intracellular virus accumulation. So it actually shows that these are productive routes, which is important. So basically what we've learned so far is these particles are taken up by clathrin-mediated endocytosis, and then they go to early and late endosomes, which is typical. Now, how about acidification?
Aha.
So acidification of endosomes is needed for the uncoating of many viruses. Early studies that they did suggested that the entry of enveloped Hep A requires acidification, but not naked. Naked HAV is very stable to low pH. Yeah, it goes through your stomach, right?
Right.
So it has to be stable to that. And so there are other markers of late endosomes that are acidified. And they can use those, and they can also use a drug, chloroquine, which Dickson knows well. This actually inhibits the acidification of late endosomes. And that's how you can ask whether you need acidification.
Poisons the P protein.
So they find naked, quasi-enveloped, but not naked, is trafficked to endosomes that have lysosomal markers. So now later in the pathway, lysosomes are fusing. And so these quasi-envelope ones seem to be going to lysosomes. Well, let's say lysosomes and endosomes fuse together. Lysosomes, of course, bring enzymes to the endosome like proteases and nucleases and things that break up membranes. Mm-hmm. So they're thinking, well, this makes sense because maybe you need to get the membrane off. Because remember, this quasi-envelope is in the endosome. It's still in the envelope. So they're thinking maybe we have to get rid of it. Yes, Dickson.
But during a natural infection, when you swallow this virus, it goes through the stomach. Shouldn't that have removed the envelope?
Yeah, probably.
So then...
But then when it makes...
may not apply to a normal infection.
No, but when it gets to your... Let's say it replicates in your gut, you get a viremia, it goes to your liver.
It replicates in your gut. Okay, fine.
Now there, maybe in the gut, it's producing quasi, in the liver, so that the blood form is protected, I would guess.
It would be nice to know that, though.
Well, the stuff in feces is naked, right?
And the previous.
So where does that come from?
From the liver, or from the gut, from one of the replication
sites. So they don't. But then it's removed as they get into the small intestine.
So one idea is that it goes through the bile duct, and that will take off the lipid. And so what we do know is that all the Hep A in feces is naked.
And the Hep A in blood in an infected individual is often enveloped.
Right, because that's coming out of the liver.
So if you got infected by contaminated blood, you'd be getting the envelope form. But you would likely have that injected and you would not eat it. Right?
Right.
Right.
But if you did eat it, the envelope would likely come
You're
off.
a vampire.
The cool thing here is if the envelope comes off, it doesn't matter.
Exactly.
Right.
It's still infectious. So this virus has the best of both worlds.
So it has two ways of getting into the cell, and it takes two different pathways once it does. Well, not different pathways. They start out the same, but it stays in the endosome longer if it has the envelope.
That's right.
Until lysosomal fusion.
Exactly.
And then that is presumably breaking down the envelope, which subsequently allows the virus to enter from the lysosomal compartment.
Mm-hmm. Mm-hmm. That's exactly right. And we're going to see a little more evidence to what's going on in these lysosomes. What else is interesting here? So the escort machinery is not only needed for the production of exosomes, but also for traffic of vesicles within the cell. And in fact, it's needed for endosomal sorting to the lysosome. So if you knock down the escort components, including a protein called ALIX, A-L-I-X, or ALI, you block the entry of quasi-envelope viruses because they can't get to the lysosomal compartment. Right? That's cool. This escort pathway, besides escort, endosomal sorting complex required for, what's the T? Transit. So don't ask me what CRISPR is. I always forget. Escort. And then they have great names for their proteins. ALIX, and another one is CHIMP. CHIMP1B, CHMP1B.
These are all in hepatocyte cultures?
Everywhere.
Or organoids.
Proteins are everywhere. What kinds of cells are they doing this in?
Mm-hmm.
We have to look it up. Some kind of cell. They're not organoids. What kinds of cells?
Because they're good hepatocyte cell cultures.
HUH7 cells, these are hepatocytes. It's hepatocyte cell line. Yeah. And what is your philosophy, everyone, on figure legends? Should the first sentence
be utilitarian? Oh, figure legends.
Tell you exactly what the experiment was done in, or should it give you a conclusion? Because people do it both ways.
Yeah.
In the figure legend?
Yeah,
I think it doesn't matter to me.
Yeah, because here it says
whether it's a title or whether it's the
conclusion,
take-home message.
Yeah, when I'm reading a figure legend, mostly I'm focusing on whether it actually explains what all the graphics are.
There are too many letters and too many groups I lose interest.
Yeah, it's
it's important to tell you what cells are being used. Now, they do here, so it's HOH75.
Right.
Okay, so, we're trafficking to the lysosome via an escort-dependent mechanism. Mm-hmm. All right, so what happens in the lysosome? So...
with these cells, these viruses also must withstand being ingested by Kupffer cells too, then. Because in a normal liver, that's the first thing you hit.
Are you ingested by the Kupffer cell?
Mm-hmm. You are.
Would all the Kupffer is like a monocyte?
It's a
liver,
fixed
right?
macrophage, basically.
It's fixed. It doesn't migrate.
No.
I don't know, Dickson. Maybe it would go to the lysosome of the Kupffer cell.
Yeah, it seems. That's what I would assume.
No.
Well, but these are also these viruses and these vesicles are circulating in the blood, so they're going to come in contact with just regular hepatocytes, right?
No, but I was thinking of the route where you swallowed it, then it went through the enterocytes, and then it got into the blood, and then it got into the liver. The moment it gets into the liver, it's going to encounter Kupffer cells.
Some of it will.
Yeah.
So the next question is, how does the particle get out of this envelope? But there are no viral proteins in it, so they can't mediate fusion. So
they can't bind to receptors.
Yeah.
With that.
So they're saying, well, maybe in the lysosome, in the lysosome there are lots
Proteases.
of proteins. There are cholesterol transporter proteins like Niemann-Pick type C protein.
Right.
There are lysosomal acid lipases.
Yes.
So they're thinking maybe these trash the quasi-envelope, so they can deplete these proteins with siRNA, the Niemann-Pick NPC1 protein, the lysosomal acid lipase, and it impairs infection with quasi-enveloped Hep A. So if you knock down these lysosomal component, but naked Hep A doesn't care.
Oh, I see what you mean. Yeah.
So they're thinking that
it's not uncloaked.
They're thinking that it's uncloaked in the lysosome, right? The cloak is a wonderful word.
Yeah. That's Alan's.
And then they do a cool, pretty experiment where they take these fluorescent dyes and put them into membranes, and they can, so they're basically staining lipids with these dyes. And so the virus has, the quasi-envelope virus has these membrane dyes stuck in it, and they can track them going into cells. And they can also use an antibody to the capsid and follow them together, right? Two different colors.
Right.
Mm-hmm. Yeah.
And it's beautiful, beautiful. And they do confocal microscopy, which in itself is gorgeous, right? And so they infect with these membrane-labeled virus, and they can show that for the quasi-envelope virus, the capsid antigen, which they pick up with one antibody, is surrounded by the labeled membranes in the lysosomes 6 hours after infection. So they say the quasi-envelope Hep A reaches the lysosome cloaked in membranes. And then, if you wait longer, the fluorescence goes away because it's being digested, I guess, in the lysosome, and you're only left with the capsid fluorescent. Isn't that cool?
Yeah.
Indeed.
I think it's a really pretty experiment. And they also have an inhibitor, by the way, of this lysosomal acid lipase. Lallistat-2. They can do this. They can show that you can inhibit the digestion or the decrease in fluorescence. So you get into the endosome, and then the membrane is digested away. So that brings the here's a cool experiment. Very cool. They take extracellular vesicles from uninfected cells. So these cells are always making extracellular vesicles. If you infect them with Hep A, they happen to contain Hep A viruses. Right. But if you take the ones from uninfected cells and you add them to cells, they get taken up by endocytosis, but they don't go to the lysosome.
Right.
Yeah.
And that's very cool because that means there must be a signal in the quasi-enveloped Hep A membranes that directs it to the lysosome. Isn't that cool?
Yeah.
It's very cool.
That's probably the coolest thing here that there is. So we got to find out what that signal
a virus-infected endocyte.
Are you confused?
No, I'm not. I'm just intrigued.
You must be learning, right?
I am. Leaps and bounds. No, I'm serious. I'm intrigued. So this is a viral component then, it sounds like, right?
Doesn't have to be.
Or when it exits a host cell and gets included into these exosomes, it's a different quality exosome than the exosomes from a non-infected cell. And those are qualitatively different.
Quality.
Somehow I think that's true, but it's not because of any viral proteins that are in that membrane. So it's hard to imagine how that is.
Mm-hmm.
Right.
Did they do a composition study on the viral membranes?
Yeah, they say later that that's in progress. They're doing some mass spec, and there are differences, but they don't know what they are yet.
Uh-huh.
But that's how you would do it, right?
Uh-huh.
You take exosomes from infected and uninfected cells, get rid of the virus,
right,
and just do mass spec on what's left.
Or even naked capsid. I wonder if naked capsid plus membrane in exosomes would give you the same result.
Separately?
Yeah, with no viral genome, but with the capsid.
So you make just naked capsids in cells. Yeah, that's a good experiment. Yeah, because that would say that any of the other viral proteins are, whether they're involved, yeah. Now, another cool experiment is...
Wait a minute, I have to go to the lab right now. I'm sorry.
Go ahead.
Yes, I'm learning.
I thought you were going to say you had to go to the bathroom.
No.
So... The cool thing about this envelope is that it makes the virus resistant to neutralizing antibodies, right? So a naked virus, you can add antibodies to it. It will block infection in some way.
Right.
But with this envelope, the antibodies can't get to the capsid, and there's no viral protein in the envelope, so you can't neutralize it. Mm-hmm. And I want to just say they use a neutralization assay, which is called a quantile infectious focus reduction neutralization assay.
Right. Say that fast 3 times.
They can't quite do plaque assays. Okay. And they have shown before that you can infect cells with quasi-enveloped Hep A and wait a couple of hours and then add antibody to the cells. The antibody will get into the cells, traffic to an endocytic compartment, and neutralize
That's weird.
the virus intracellularly.
That's weird.
And that's, as Kathy knows, that's been done with adenovirus too, not quasi-envelope, but just regular. You can get this post-entry neutralization in an internal compartment of some sort. So if you add this inhibitor of the lysosomal acid lipase, this drug, it now blocks neutralization by that mechanism because it's not right. Because now it's not opening up, and because the neutralization by those antibodies added to cells depends on the membrane being removed in the lysosome. That's a cool experiment too, right?
Mm-hmm.
Yes.
Okay, so basically, let's see. So far, we have endosomal uptake. You go to lysosomes, the membrane is taken off. And now we have a capsid in there. And somehow these quasi-enveloped are getting directed to the lysosomal pathway, but we don't know. We don't know how that's being done. I think the other question here is now, if in a late lysosomal compartment we have these naked particles, the quasi-envelope has been removed, how does the RNA get out? Right?
Right.
It's still in a stable shell.
Or why don't the cathepsins digest the capsid?
Say that really quickly.
Why don't the cathepsins digest the capsid?
In the lysosome?
Yeah. Cathepsin D is a protease. It should be zapping them like crazy.
Well, what's the specificity of a cathepsin? No idea?
No, I don't think it's very specific because anything you eat via the lysosomal pathway gets digested. It's degraded. I mean,
I don't know the answer to that, but it's not, obviously.
Right.
But somehow the RNA gets out of the capsid into the cytosol. That's where it has to go, to the cytosol. And we actually do not know how that happens. Now, poliovirus can induce pore formation in membranes. And that's how we think the RNA gets out. Okay? And so they're saying, well, does Hep A make pores? So they do a great assay. So they have a ribotoxin.
I love this one. Yeah, this is nice.
Which is membrane impermeable. So this inhibits protein synthesis, right?
Right.
They can load this into cells. It gets incorporated into endosomes. and it will stay in the endosome unless something pokes a hole in the endosome. Then it will get out and will inhibit translation, which they can measure, right? So, you look at global protein synthesis. When you infect with quasi-enveloped Hep A in cells that have this ribotoxin in it, you get an inhibition of protein synthesis. So somehow the
virus facilitates the
there's damaging the lysosome.
Sure.
So that the ribotoxin is getting out. And maybe that's how the viral RNA is getting out of the lysosome. But naked virus doesn't do that. Naked Hep A doesn't do this. It doesn't seem to damage endosome. Remember, the naked never gets into the later compartment.
So do you get a partial fusion then of the exosomal of membrane with the lysosomal membrane?
I'm sorry, for lysosome with endosome?
Yeah.
Yeah, well, that's part of the pathway. As the endosomes come in, they fuse with lysosome.
So this has a portion of that membrane on itself. So you're going to get a hybrid membrane, basically. Then maybe it's looser junctioned at that point and it lets the stuff out. I don't know.
I'm not aware of that sort of thinking.
Or do you mean as it
fuses with the lysosome?
Does the viral envelope fuse with the lysosomal membrane?
That is what I'm asking. That is what I'm asking.
Well, no, what they think here is that the viral membrane is digested in the lysosome.
Right.
It's destroyed.
And then something associated with it is going to break a hole in the lysosomal membrane and get the virus into the cell.
And somehow the envelope virus is injuring the endolysosome.
By the act of being digested?
Sorry?
Well,
by the act of being digested?
Don't know.
I mean,
yeah, maybe
there's a missing step here somewhere, isn't there?
Yeah, well, there's so you've got some viral particles inside this vesicle,
right?
That's initially an exosomal vesicle floating around in the bloodstream. It's shielded from immune surveillance there. It gets ingested into the cell in the endosome. And I'm thinking there may be some viral proteins from the previous infected cell tucked in there with the virus in the vesicle. And those could be something that causes this, because we know there's something the virus does that's directing the trafficking, and there's something the virus does that's going to poke a hole in the lysosome. And I'm thinking there are probably some additional proteins that the virus has packed into its vesicle and brought along for the trip.
Well, you can also think about the HPV story where there's that little tail of, peptide that sticks out and then allows it to pull through.
Yep.
But I was going to put on my grant reviewer's hat and say that in addition to showing this damage to the endosome by the ribotoxin experiment, they show rigor by using a different assay, which is their dextran assay.
That's right. Right. They show rigor. That's something you need to now comment on in grants, right? And you have to show how are you going to show experimental
multiple ways of showing the same.
So you can load the cell with these fluorophore conjugated dextran particles or dextran, and you could, since they're fluorescently labeled, you can see them and count them, and they see that when you infect with envelope, but not envelope Hep A, the number of fluorescent punctate things in the cytosol are decreased because they're being released from the lysosome, and then they're diffuse and you can't see the fluorescent as much. Yeah, it's cool. It confirms it. Mm-hmm. So basically, this quasi-envelope virus encodes in the lysosome. It somehow causes membrane damage. We don't know how. It's a good question, and that's the timing of that is the same time when the genome would get in the cytoplasm. But they don't know how that happens. Now, the non-enveloped enters the cytoplasm earlier. In fact, it starts its protein synthesis earlier. But we don't know how that happens either, because those are not damaged, apparently, by the virus there. And they say maybe we just couldn't detect it. Maybe it's reversible damage, but somehow the RNA's got to get out, right?
So they can label the capsid protein, of course, with a fluorescent dye, and then just see when it disappears.
Well, they did it other ways. They use antibodies, and they know that the non-envelope, the viral RNA is getting out earlier by measuring protein synthesis.
And they can say where it's getting out?
They can say that it's getting out in the endosome before it fuses with the lysosome.
So early.
Yes, earlier. And the kinetics of replication are quicker too.
Okay.
They're consistent with that. And the envelope goes later. It takes a little bit longer for translation to begin. Yeah.
So what happens to the capsid then when the non-enveloped naked capsid virus gets into the early endosome?
What happens to it?
Mm-hmm.
Well,
does it get digested?
So what happens is that, yes, so the RNA comes out, and then those endosomes, which have the empty capsids in them, go through to the lysosomal part, and they get digested.
You mean so there's a little hole in the capsid which allows the RNA out?
Presumably.
Wait a minute.
In polio, you can see the hole, for sure.
Could you do cryo-EM and see this?
Well, you can do, yes. So it's been done for polio. I don't know if we did this paper. I think we did, showing this umbilicus.
Yeah, no, I remember
between polio
that's
and the membrane.
Yeah, that's right.
And so you can see this by cryo-EM, yes, totally. But they don't have any evidence of this.
So maybe that's an addition that Kathy's grant reviewer hat would like to ask.
Oh, yeah, make them do cryo-EM on top of it.
Right.
It's an easy thing to do. You just freeze it and look.
Maybe this is why the paper's in eLife.
Yeah. Ooh.
One thing about the paper being in eLife is that everybody has access to it.
So
and there's a nice model figure at the end of Figure 5 that I've just been following along.
Yes.
Yeah, it's a beautiful figure. Yes.
Yeah, very.
And then
nice
some of the other
surprises.
ones, like the ribotoxin figure, that's part of one of their figures. They have a little cartoon for that, and they have a cartoon for another figure. So it's nicely graphically presented.
Mm-hmm.
Yeah, they did good illustration.
What country just recently banned Elsevier's journals?
It was University of California system.
Right.
Which could be a country.
You can call that a country.
Yeah.
Because the trend is moving in that direction, isn't it?
If you see is not a trend, is it? It's just one state. California is always...
and they didn't ban them. They refused to subscribe.
Yeah, well, that's...
You can still read it if you
about
want.
the same thing, right?
If you're at Starbucks, they'll let you read it.
They're not going to spray you with pepper spray for bringing it on the Berkeley campus.
This is true. Okay, fine.
And I think that's all we need to say. There's one other experiment, but it's kind of obscure and not necessary to. So this shows that the quasi-enveloped and the enveloped have two ways of getting, and they both come in by clathrin-dynamine-dependent endocytosis. Somehow the naked gets out of the endosome early. We don't know how that happens. Maybe finding a proteinaceous receptor will help to figure out how that happens. Mm-hmm. The quasi-envelope continue on, the lysosomes fuse with those endosomes, and then the enzymes in the lysosome digest the envelope, and somehow that damages the lysosome, endolysosome, and the RNA gets out, but we don't know how either.
So I'd like to suggest a title for this show right now, and it's called Either Way You Lose.
Who's the you in that? The human host?
The cell,
human
that's
host.
exactly. Either way.
Either way. Is that a takeoff on some other?
Probably.
Either way you lose. So here's what they say. Quantitative proteomic studies provide some support for the hypothesis that there's a signal in the envelope of the quasi-envelope virus that directs them to lumen to the lysosomes. But what it is exactly, we don't. So that means there's a difference between the membranes, but we don't know what.
Right.
And Dickson's naked capsid experiment would be a start on figuring out if that's a capsid protein versus a viral protein.
It's a protein.
Yeah.
And by the way, no other picornavirus is known to be trafficked to the lysosome for uncoating.
Wow.
So here's my question to you. Mm-hmm. What other virus outside of the picornavirus is trafficked to the lysosome for uncoating? There's more than one.
You're asking the wrong person.
I mean, it could be anyone. Listeners, shouting.
Listeners are shouting it out.
Vincent
right now.
looks at me and says, "Like, I
well, you
want
happen
you
to be
answer."
here. You happen to be here.
I wish I could answer that.
So,
question.
reoviruses are one.
Rio? This is an RNA virus, right? And it's in the lysosome, right?
Right.
And there are RNA aces in the lysosome, right?
Mm-hmm.
So that means that this virus doesn't come in contact with the RNA aces. So it probably is the fact that the capsid sits up next to the membrane to let the virus out in both experiments.
Probably, yeah.
Probably.
That would make, because you don't want the RNA to come out into
Ever.
lumen, because it'll get digested. Absolutely. So it probably abuts the membrane and gets across, which, says, man, there must be a proteinaceous receptor or something holding it on.
Yeah, my money's on both of them getting out the same way once the
I would agree.
envelope ones lose their envelope.
That makes sense. That does make sense.
That's parsimonious.
There you go.
It's also simpler, right?
Yes.
That's also Occam's razor.
Right.
So
I shave with Occam's razor.
What was I going to say? Yeah, so reoviruses, they stay in until the lysosome, and then the lysosome strips off the outer shell, and the inner shell particle is hydrophobic and pokes through
There you go.
the lysosome membrane.
Wow.
And as Kathy said, the adenoviruses have a protein that gets them out of the endosome, makes holes in the endosome. But we're not seeing any holes here, but as they say, maybe we're just not quick enough to see them.
Right.
So in light of there being a receptor, so for polio, we know the receptor triggers this uncoating. It makes the massive changes in the particle, which then form a pore in the membrane, between the capsid and the membrane. They say there are some monoclonal antibodies that bind the hep A capsid very strongly, and they destabilize it. And they say maybe this mimics what a receptor could do, destabilize it so the RNA could get out. Right?
Is the viral genome under pressure inside the capsid?
Probably, yeah.
So it's like a little pump.
It's under enormous pressure. Under pressure. There was a song called Under Pressure.
Yes.
Right?
I know. ZZ Top.
Was ZZ Top, really?
Yeah.
I don't think so.
I thought it was,
she's got me under pressure.
No, that's not ZZ Top.
Yes, it is.
Here, under...
David Bowie.
Yeah, David Bowie.
Okay.
Yeah.
She got me
Queen.
She got me under pressure.
Queen
and David Bowie.
They both did it.
Well, ZZ Top uses that same phrase too. I'm not kidding.
Pressure pushing down on me.
Oh yeah, that's right.
And it starts with mum, 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 right? Right, Kathy? Where'd you go?
Oh, sorry. I was muted.
Oh.
Maybe. I was thinking of a different song about pressure that I thought was by an Australian group, but...
So now we have a connection between this TWiV and the last one. We have Queen. It bridges both, right?
There you go.
Mm-hmm.
Well...
Anyway, those are two cool papers. Well, the mitoviruses in general are very cool. And then the quasi-enveloped. And remember, other viruses do that as well. So this is not unique to hepatitis A virus. Okay, let's read some emails. Galen writes, "Hello, TWiVers. I'd like to present this paper for the TWiV team to consider presenting on a future TWiV, as it reports on a particularly extreme example of a category of virus I hadn't previously heard of, the multipartite virus. Surprisingly, this publication came to my attention when my mother, who is distinctly not a scientist and usually barely tolerates when I talk about my own work, sent me an article she stumbled across in The Atlantic. And this, of course, is a paper we talked about last time.
Yeah, the plant virus.
Over the last several years, I've come to really enjoy the TWiX cohort. I like that, the TWiX cohort. And as I am now a second-year micro-grad student at the Oregon Health and Sciences University, these podcasts have certainly helped me think about my own work and have inspired consistently interesting discussions. I'm working to characterize ways in which host lipid factors play roles in Mycobacterium tuberculosis infection. The dulcet tones of the various TWiX team members help keep me happy and thereby uninfected in the BSL-3. Please keep up the good work, and I look forward to continue hearing the weather reports." Well, hope you enjoyed TWiV 539.
Yes.
Because that's where we did it.
Yeah.
So this next one is Peter Hotez being interviewed by Joe Rogan. Now, some of you listeners may know Joe Rogan, or Rogan. I don't know how you say it. But this is an hour-long interview of Peter Hotez.
Right.
And it's an interesting format because he sits across from you at his desk, and there is one camera shot from the end where you can see both people facing each other, but his desk is covered with crap. All kinds of, tchotchkes that people have in their office. I have a lot of stuff on my desk. I have a lot of viruses.
Not a lot of stuff.
But he has all kinds of stuff. So, it's interesting. He talks a lot. Thank you.
A lot too,
Thank you, Martin. Dickson, can you take the next one?
You bet I can. Sam writes, Dear TWiV team, your discussion in episode 539 of plant grafting reminded me of my favorite science joke. Now that I'm in law school and no longer surrounded by scientists, I never find myself in the company of people who would understand it, so I thought I'd share it with you. Question. What was the greatest biology experiment of all time? Answer. When Luther Burbank crossed the Rocky Mountains with his wife. Thanks for all. That's right. Thanks for all the work you put into the podcast. Cheers, Sam.
Look,
he's a
law student.
Law student at Columbia. P.S. I also wanted to submit a listener pick, but because it's a shameless self-promotion, I wanted to leave it to your discretion whether it was appropriate to mention on your show. The Columbia Science and Technology Law Review, an academic law journal staffed and edited by students at Columbia Law School, now has a podcast. STLR, pronounced stellar, Conversations. This podcast will explore the intersection of science, technology, and the law, and will feature a mix of in-depth interviews with authors published in our journal and surveys of legal topics from industry experts. Our first episode, released earlier this month, discusses a law most people have never heard of, Section 230 of the Communications Decency Act, but is ostensibly responsible for allowing the internet to exist. If you've never ever wondered why Facebook doesn't get sued every time someone posts something illegal on the site, for example, bogus medical advice, this law is what prevents that from happening. Later this year, we will have an episode about discarded DNA and its use in criminal prosecutions. Our objective is to prevent complex legal That's
present.
What it says.
I think they mean
it says prevent. I'm sorry, I'm going to read it. He's a lawyer. He knows what he's talking about. Our objective is to present, I'm sure he meant, complex legal topics in a format accessible to a lay audience. We are available on iTunes, Google Play, and Spotify, as well as our own blog, stir.org/podcast.
I could listen to that.
You bet.
I bet Trudy would like it too.
You're prevented from doing that. I'm sorry.
Don't you think? So this law that prevents Facebook from being sued, I think we should change it.
I don't think I'd want to go down that road.
Why?
No, this is a kind of a don't shoot the messenger thing.
Well, I think we should sue Facebook.
I'd like to know where the messenger comes from then.
So they're agreeing to, what are they agreeing to lately? They're going to stop the
anti-vaxxers.
Anti-vax.
Right.
Kind of don't believe.
Well, I think they should identify where they come from, and if they do that, then you'll just do it under a pseudonym. So there's no way.
Is it a good law to have? You're saying?
Yeah, I think this law is not the problem.
Is it a freedom of speech kind of thing? Because
I
Facebook
think
is. No one said Facebook should exist.
But that doesn't mean that the people who post bad medical advice can't be sued.
People themselves, not the
not the
site. Oh, okay.
Not the medium, not Facebook.
I think if you run a general knowledge internet kind of a thing like Facebook, you should have some say as to what goes on it.
Oh, they have say.
Like
they can ban whatever they like. They ban all kinds of things. They'll take down a picture of somebody breastfeeding a baby.
You're
That's
right.
gone in an instant.
Yeah.
But the Nazis and the vaccine deniers, that stays up.
Well, no, they've taken down the Nazis recently.
They're starting to take down the Nazis, but those are editorial decisions by Facebook.
No, I agree with you, and they're random discriminatory.
But I think if you say you're going to repeal this section of the Communications Decency Act, you're going to open up a whole can of legal worms. I would have to listen to this podcast episode and or talk to other lawyers to find out how many worms are in that can. But I know there are some ones you don't want to deal with.
Okay, we'll leave it.
Yeah.
No problem. Good. But this is, I'm going to listen to this podcast. By the way, I gave a lecture in an immunology course at Columbia last fall, and Sam was in the audience.
Yeah.
That's why I know Sam, yeah. He was taking the course. I said, "Are you going to take my virology course?" He says, "No, I don't have enough time." Well, too bad. Alan, can you take the next from Anthony?
Yes, Anthony writes, her son died, and then anti-vaxxers attacked her. And this is an article from CNN, and it's about this woman who I just started reading the article. She's lost a 4-year-old son to flu, and found a bunch of hateful comments from anti-vaxxers. Yeah, this is just, people are awful.
Correct.
Yep, especially under the anonymity of a website, right?
Yeah.
Oh,
okay.
He continues, yeah.
Right. So continuing with Anthony's letter, the Washington Post has an article on Amazon removing anti-vax titles, but as I've used up my free access, I've not read it yet.
Right.
And another article, Kentucky governor, this is the one I think we talked about already, the Kentucky governor boasting about taking his children to a chicken pox party. The character should have been committed, not elected.
Here.
Why isn't the intentional exposure of children to disease prosecuted as abuse? I think it ought to be, but I'm not the one writing the laws. So
there's a good editorial in Science, I think this or last week, by a law professor at UC, and she says there is legal basis for prosecuting in some of these cases, especially at the state level. She says states have a legal obligation to protect the health of their constituents.
Of course, if you're the governor of the state, that probably provides some insulation.
He is the governor, isn't he?
Yes.
Kathy, can you take the next one?
That's Islam writes.
Yes.
Dear TWiV team, I just wanted to tell you a quick story. My wife, Rhonda, is a preschool teacher and curriculum coordinator at a private school in Massachusetts. She has a master's degree in early education and is always willing to try new fun things in her class. She was curious about the book that I was translating to Arabic, Paul Has Measles, which you guys talked about previously in multiple TWiV episodes. She passed by my desk last weekend and saw the lovely illustrations on my computer screen. Are you reading a book for kids? She asked. No, I'm actually translating a children's book about vaccines into Arabic. She flipped through the pages of the PDF and asked if she could read it to her students. Absolutely, I said. I gave her the virology.ws link, and the next day she sent me pictures of her students reacting to the book's content projected on a big TV screen. This was the cutest thing ever. Kids really loved it and wanted to spend more time going over this interesting material. So she decided to print and laminate the book, which is now available for the kids to touch, read, and ask more questions. She was amazed by how engaging the content was for them. It seems that this experiment is worth replicating with other scientific books geared toward this young age. I thought that this story could inspire some of the TWiV listeners to do the same. If you are a teacher or know one, please don't hesitate to spread the word. Thanks, Islam.
Nice.
That's great. It's really great.
I guess because the story involves kids, right?
Yeah.
Getting measles. That's why they like it, right? It's not like a peripheral thing that they can't relate to. Yeah.
Well, it involves kids. It's a well illustrated book. It's written in language that they know, and it's just a really well done production.
Yeah, it is.
And the cartoons are great.
Yeah.
So next, Bob writes and sends us, in view of your TWiV 503 on citrus greening, I thought you might find interesting this astounding story. He sends a link to a Nature news feature. It's called Antibiotics Set to Flood Florida's Troubled Orange Orchards. So we talked about citrus greening on TWiV 503, and this is a bacterial disease. And our host... I forgot her name.
Simon?
The other one.
Oh, not Ann Simon. We did this last week.
Yeah, we did.
Yeah.
Well, let's just look up TWiV.
Folimanova.
Keep mixing up these plants.
Folimanova. Yes. Svetlana. She told us about this. She said it was going to wipe out the citrus in Florida. Yeah. So they're dumping tons of antibiotics. Streptomycin and oxytetracycline. And so some people are worried about this because they think it's going to screw up the microbiome, and others say don't worry about it. So, but they're going to do it because there's not much else to do, right?
Arg.
Yeah, so they're saying they could spray 440,000 kilograms of these drugs on the orchards. And This is, by the way, the bulk of antibiotics that are produced in the world right now do not go into humans.
Oh, that's right.
And in fact, most of them are going into farm animals in developing countries.
Right. We've actually stopped doing that.
We've stopped doing that. Europe has stopped doing that. But now in the developing world, most of the antibiotics are going into farm animals. And now apparently we're going to be spraying them on the orchards, which it really seems like a bad idea to me.
But Alan, don't you think that was a way of dumping antibiotics that we could no longer use in our own country? We could dump them somewhere else?
Well, no, there's a market, so it's being fed, and a lot of those are being manufactured locally. I mean, these are many of these are way off patent.
Okay.
And the profit margins on drugs for agriculture are tiny.
Agreed.
So this bacteria can't be cultured, so you can't tell if the antibiotics are going to affect it. They have to depend on field studies, which they say are equivocal. Don't know.
Yeah.
One group is going to be extremely happy, and that's the California citrus growers.
Well, until this thing shows up.
Exactly
there,
right.
yeah.
No, exactly right. But
I mean, I agree that it's important to keep the crops growing, but I don't know.
At what expense?
Yeah.
And then when it rains and it gets washed down into the Everglades, that's lots of luck.
Right.
Yeah.
Okay.
What could possibly go wrong?
No, that's right. Go wrong. Go wrong. Go wrong.
I hear that all the time these days.
Yeah. How bad could it be?
How bad could it be?
That's my wife's favorite expression. Then we find out that was much worse than I thought.
We used that last week when we talked about the Australian.
Right. Yeah. Mm-hmm.
What could go wrong?
That's right.
What could go wrong?
All right. Let's do some picks then.
You got it.
Alan, what do you have?
I have a useful travel guide. Warning, trigger warning.
These are
click on that
angiosperm things, yes?
Yeah, this is places to go and to see the beautiful displays of cherry blossoms that are just coming into bloom now, all up and down, especially the East Coast. Not just the ones at DC, but all over the country and up into New England. And for Vincent, this is a list of places to avoid for the next couple of months.
Months? Is it that long?
Some of them toward the north, I think, are going to go into May or early May. Some of them have happened already. The Cherry Blossom Festival in DC, I mean, because of climate change that keeps happening earlier and earlier in the year. And they've had years where the Cherry Blossom Festival happens and the cherries have already blossomed and done. But
is it ongoing right now, Alan? In Washington?
It is.
Last week of March, first week of April.
Really?
Sometime in there. And they only bloom for a couple of weeks, so it's sometime in that window of time.
Right. Be down there.
And there's a gorgeous slideshow of 25 pictures of cherry blossoms.
Cherry blossoms in different contexts, including the base of Mount Fuji.
New Haven has some.
Has some. Yes, I remember those.
Brooklyn Botanic Gardens has a wonderful display. It's just absolutely gorgeous.
Yeah, so that's actually, what's the date they give for that one?
Late April.
I think, it was in here last time I looked at this article. Anyway.
Vincent, didn't I send you some artwork of the cherry blossom viewing parties?
I think so, yeah.
When I was at the Chicago Art Institute. Yeah, because
here it is, April 27th to 28th is the
late April. That's right.
Sakura Matsuri at the Brooklyn Botanic Gardens.
Exactly.
So there you go.
All right. Enjoy, everybody. Enjoy your cherry blossom. Dickson, what do you have?
Well, I have an article that once again confounds the people who are deniers for climate change. Another dead zone is viral, as I indicated, but it's an article documenting what's happening right now in the Midwest. Kathy, you're just on the cusp of all that, but I'm sure you're hearing about it anyway because we're having a record melt and rainfall in Nebraska, all the way down through Missouri, all the way, and it's eventually going to reach, of course, into Arkansas and Mississippi and Alabama and Louisiana. It's the worst flood that they've ever had. And the worst flood that they ever had before that was in 2011. And that was called a 1,000-year flood. And now this is the second 1,000-year flood that they've had in only 8 years. So that's going to tell you something. They'll have to redefine this some way. Ag losses are in the billions of dollars right now. And they said if winter wheat stays underwater for more than a week, it dies.
Why does it say this governor of Missouri called President Obama and received?
This is an
I
old
don't
article.
know.
You've linked to an old article, but there is a current event going on about this as well.
Is that not a new article?
Apparently not.
No,
I can't
December,
find a date.
All right,
I can't find a date either. I've got December 30th.
I'll change the article because I had another article there before that. But when I ran across this one, it looked like it was much more. You know what? I'm going to put them both there because they're both saying the same thing.
This must be
except
December
at different times.
At different times, exactly. This was something that happened a few years ago.
No, 2011. That's when it happened.
2011, and now 8 years later, we're having another 1,000-year flood.
My mistake.
So
no worries.
Your story is accurate. This story that you linked to happens to be an earlier. This happens to be the previous one. It's easier it's easy to get your 1,000-year floods mixed up when they happen. 8 years.
Time flies when you're not having fun either.
Yes.
Okay, Kathy, what do you have?
I picked something that relates to Peeps. And so for those people who might not know about Peeps, those are the marshmallow treats that are, in this case, they're rabbit ones. And so this is a link to someone's Twitter thing. And even if you're not on Twitter, you can just see the initial thing. And what he's done is he's got pairs of the rabbit Peeps, and he's injected them with a bunch of different fungi, and then just gotten all kinds of responses to that. And so you can see the Peeps, and then people are suggesting other ones to add in round 2, and people talking about whether, they're too dry and what's going to happen to these fungi. And it's just really fun.
One of the fungi is Cryphonectria parasitica, which has a mitovirus.
Yes.
Right. Yeah. I didn't even think of that. Yeah.
Yeah. So he's testing the susceptibility of Peeps to various fungi.
Right. And so, and people are suggesting all different kinds of permutations and stuff. And the one that I didn't read through all the comments, so probably somebody already made this one. But my thought was, okay, he's injecting these rabbit Peeps, but what if he did the chick Peeps? We could know something about the bird Peeps versus the mammal Peeps. So,
yes, Peeps. That's right. It's not just.
Still no official comment from Peeps brand. I suppose this was not the best way to pursue a sponsor.
Not a peep out of them.
It's not just these Peeps is you tell
but
people, but they're these things too, right?
Yeah.
They are somehow linked into it because their logo shows up of either people that liked it or that retweeted it or something. Peeps brand is there. So, yeah,
cute. I used to love Peeps.
Mm-hmm.
Peeps can also mean people.
Good.
Yeah, I use it that way too.
Hey, Peeps.
My wife, her mom is Episcopal, her father's Jewish, and she likes to, every Easter, she likes to have Peeps on matzah.
Wonderful.
Little snack.
Ecumenical. All
right, my pick is another Ars Technica article by Beth Mole. Emergency declared in New York over measles. Unvaccinated barred from public spaces.
Good idea.
County official calls resistance to outbreak response unacceptable and irresponsible.
That's Rockland County.
Rockland County. They have a huge measles outbreak.
They do.
So effective midnight this past Wednesday, anyone 18 or younger who has not been vaccinated against measles cannot be in public spaces.
How do you prove that they haven't been vaccinated?
I don't know. That's a good
they have
question.
Spots.
Yeah.
Stop it. They don't have
Well, no, I mean, this is something you would enforce. You would
do a spot check
after.
You said they should do a spot check.
They do a spot check.
That's what they do.
This is something you would enforce after somebody shows up and spreads measles to a bunch of other kids at the school, and then you would say, okay,
it's true.
You were in violation of this. It's not something where you're going to be able to go around and ask for people's vaccination cards.
No.
But it puts everybody on notice that this is something you could be prosecuted for.
So if you want to learn about the outbreak and so forth, it's a good article. So we have one already listener pick from Sam, his podcast. That reminded me.
This is very good, by the way.
So Sam's podcast is on the intersection of science and law. Now what someone needs to do is the intersection of science and art. But I'm sure maybe someone does it
There's
already.
a lot of that out there.
Podcasts?
So, not podcasts necessarily, but
they want a podcast.
Lots of information.
Sit comfortably in my car and listen.
Yeah.
By your fireplace.
Wait, but there's
By the
something about
fireplace.
the idea of listening to something that's something that you
Art.
ordinarily
Should
will
be C.
Yeah.
That's correct.
So if they do it about music as
Right.
an art form,
That's
could
true.
Well, they could talk about it, right?
If
Right.
they're florid enough in their description, you
Like
can
we-
actually visualize it.
We talk through figures.
We
and
have to talk
papers.
to figures,
Yeah.
We do,
you know.
we do,
Yeah.
of course.
True.
And we also have another listener pick from Josh who writes, I was reviewing visualizations at d3.js and thought you guys might enjoy this. I am incapable of checking this for accuracy of representation, but I believe the code below the picture could be employed to accurately present the data if it is not already accurately displayed here. So this is a tree of life,
Right.
phylogenetic tree, inspired from a figure from Nature and Jason Davies.
Oh, and as you move the mouse around,
Yeah, it lights up the relationship.
it lights up the path, the evolutionary path, tracing it back to
Exactly.
the origin of life, I guess.
Yeah.
Or
Yeah.
and it's pathogens all around, right?
No, not all. We're
No,
there.
you got mice.
Actually, yes, because we're there too.
Yeah, we got
Well, yes.
musculus, Homo sapiens.
Oh, yeah, okay.
Oh,
A lot
I
of
see.
pathogens.
You know, right next to us, there's Giardia.
But you don't see any of the nodes, though.
No.
It's kind
It's
of
a
interesting.
simple-
And then the code for it is below.
Mm-hmm.
Dickson, does the code look good?
How would I know?
It runs.
Exactly. Yeah, it works.
It's my usual standard.
That's it.
Wow.
Cool.
Yeah.
It's pretty
Yeah.
cool.
Sweet.
What is it that he mentioned? The D3.js?
Some kind of
D3.js. JavaScript? Oh.
no. I think it may be a
library.
conference.
It's a library. A JavaScript library for producing dynamic interactive
Okay.
data visualizations in web browsers. There you go. That's how it was
Okay.
done. JavaScript. Cool. Thank you, Josh. And that is TWiV 541. And you can find it online at microbe.tv/TWiV. We don't have any JS there, but there's nothing generated on the fly.
BS.
Really? There's probably it's a WordPress site, right?
Well, I guess it is generated on the fly.
Yeah, I'm sure there's JavaScript on the page somewhere.
Yeah, it is. Yeah, it's not a static
Right.
HTML page. It is all WordPress pages are generated on the fly. Right?
Mostly by PHP, but I'm sure there's some JavaScript anyway.
So there you can find show notes if you want to read some of these letters or find links and so forth. But mostly you'll listen on your mobile device, phone or tablet. And if you do, please subscribe so we know how many people are listening. That really makes a difference for us. Send your questions and comments to TWiV at microbe.tv. Please, if you want to support us financially, go to microbe.tv/contribute. We'd appreciate it. Dickson Despommier is at fortimopolis.com and thelivingriver.org. Boy, I have such a choice every week. Thank you, Dickson.
I love it when you talk that way.
Thank you very much.
Oh, it was a pleasure.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at turbidplaque.com. And on Twitter, he is a really interesting handle, Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
Unexpectedly direct.
Yeah, it's a good handle to have, Alan Dove.
It's a good one.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support, and Ronald Jenkins for the music. Been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.